Hello and welcome to today's webinar featuring Brian O'Rourke. I'm Claire Cameron, Engagement Editor at Spectrum, the home for autism research, news and analysis, previously found on Severity.org. We'll begin the presentation in a moment, but first, know that we'll be fielding questions at the end of today's presentation, but you can ask them at any point during the session. To ask a question, submit them via the chat window to the left of your screen. And as always, I'll note that if there are any members of the press tuning in, you can report only information presented during this webinar if that material has already been published elsewhere, or if you have first obtained express written permission from the presenter. Brian O'Rourke joins us today from Portland, Oregon, where he is an assistant professor in molecular and medical genetics at Oregon Health and Science University. Dr. O'Rourke earned his PhD from Yale University's Department of Genetics in 2009, working in Dr. Matthew State's lab. He then went on for postdoctoral training at the Department of Genome Sciences at the University of Washington under Drs. Evan Eichler and Jay Schender, before joining OHSU in 2013. Dr. O'Rourke's overarching goal is to understand the molecular basis for neurodevelopmental disorders and translate fundamental insights into meaningful clinical interventions tailored to an individual's genetic makeup. Over the last decade, his pioneering work has focused on developing and implementing new research paradigms and technologies to unlock the genetics of autism. Dr. O'Rourke has received a number of early career awards, including the Alfred P. Sloan Foundation Fellowship in Neuroscience, the NARSAD Young Investigator Award from the Brain and Behavior Research Foundation, and the International Society of Autism Research Young Investigators Award, and the Clingston-Simons Fellowship in Neurosciences. We're very grateful that Dr. O'Rourke has agreed to present some of his work today. Welcome, Dr. O'Rourke.
Thank you, Claire, and thank you for Spectrum for having me. So today what I thought I would do is try to give everyone out there listening to the webinar a big picture view of how really there's been a quick acceleration in the pace of autism genetics these past several years, and I'm using this title from 0 to 60, meaning basically going from 0 high confidence genes for autism to now 50 or 60 in just a few years. So the way I'm going to structure my talk, I'm just going to do a few slides to just really highlight how new technologies have really revolutionized our ability to do genomics. Next, for the meat of the talk, I'm going to tell you about how we've tried to use these new technologies with new approaches to actually go from not just finding candidate genes that contribute to autism risk, but actually starting to move into what I'm calling a new era of known genes for autism risk, where we have many, many high confidence genes that we are confident that when you disrupt them may lead to autism in a majority of circumstances. And finally, I'll touch a bit at the end, a few kind of new frontiers in autism genetics and where the field might be going the next several years. So first, the genomic revolution. So most of you probably know that the first draft of the Human Genome Project came out in 2001. It actually came out when I was taking advanced genetics as an undergrad at Fresno State, and we actually read the paper, and it was really exciting. And since then, the price to actually sequence 1 million bases of DNA has had a precipitous decline. And so what I thought I would do is just take you through some kind of real-world examples, just to give you a sense of how this has really changed, at least in my career. So starting back when I was an undergrad in 2001, driving this kind of beat-up used car, to sequence 1 million bases of DNA cost somewhere around $5,000 or $6,000. We fast forward to the end of my graduate work at Yale, and you could sequence 1 million pieces of DNA for about the price of kind of a second generation iPad, about $500. Fast forward just 2 years to when I went to start my postdoc in genome sciences, that price had dropped to about $1, so about the price of a Snickers bar before kind of chocolate price inflation. And now here in 2015, 2016, in my own lab, we can actually sequence that same million bases for about a dime on a sequencer that sits on a benchtop in my own lab. So you can imagine that this rapid drop in cost can actually lead you to ask new scientific questions that you couldn't even have dreamt of 10 or 15 years ago. And really what's made this possible is we've moved from a paradigm of Sanger sequencing where we were sequencing kind of single molecules one at a time in a kind of serial manner to this kind of new era of massively parallel sequencing where we can sequence millions of these bits of DNA all at once in various devices. The other big revolution that's really allowed much of the work that I'm going to tell you about in the rest of the talk is what I'm calling the Exome Revolution. So the exome is simply all the protein coding exons in the genome. And in genomics, we have to add -ome to everything. And so all the exons in the genome became the exome. And this is really a new term. So this term really didn't exist until kind of early 2009 when Sarah Ang, who was a graduate student in genome sciences in Jay Shenduri's lab, published some seminal papers showing that kind of using existing technologies on a single microarray, you could actually capture an entire exome and then sequence it with these next generation massively parallel sequencing technologies. And really that was the impetus for me wanting to go to the University of Washington, to the Department of Genome Sciences, to actually try and leverage that technology. So you might be asking yourself, if you're not a geneticist or genomicist, what's the big deal? Why can't you just sequence an exome? Why is it so difficult? And that's really because of how our genome is actually organized. So I'm showing you here kind of a cartoon diagram of a gene from the Human Reference Genome Browser. And you can see here these exons are these kind of fat chunks that I've highlighted with these arrows here. And you can just tell by looking at this one gene, CHD8, that these exons are scattered. So they're not in kind of large, discrete chunks. And if they were, it would be fairly trivial to sequence them because you could just amplify those chunks and sequence them. And it gets even more challenging given that there's something like 20,000 or more genes, 250,000 of these protein coding exons, all scattered throughout the genome in different sizes and in different distributions. And so what this exome technology really allows us to do is sequence these exons, which we think are the most important parts of the genome because they're what gives the instructions to code for proteins at really a fraction of the cost relative to sequencing an entire genome. So the exome is about roughly 1% of the entire genome. So you can sequence just that 1% rather than sequencing the rest at far reduced costs. So those are really the two big revolutions that have allowed us to make rapid progress in autism genetics. And now I'm going to tell you kind of two connected stories about how we've used these technologies to first find candidate genes and then actually validate them. So first, just very briefly, since this audience I'm sure knows a lot about autism, I just wanted to make a few points. So one is that currently we don't have any kind of medical test for autism, and that can make doing genetics challenging if you're not sure who's affected and who's unaffected. Currently we still have, I would say, very little biologic understanding of what the underlying causes of autism is. But we've known for a long time that autism has a strong genetic contribution, and that's come from both twin studies and family studies looking at siblings and other relatives. But we've also known for a long time is there's no single autism gene. So it's not like Huntington's or cystic fibrosis or other genetic disorders you may have heard of where there's a single gene of major, what we call major effect, that when you mutate it, leads to autism. So we call that fact genetic heterogeneity, which just basically means there's a bunch of different genetic factors that are likely contributing to autism. And that genetic heterogeneity has really made things difficult as far as finding new genes for autism. And what we've been kind of had in the field, at least when I began my career, is many candidate genes, but really few known genes. So we'd have lists of 100 or 200 genes where there may have been a family or two that had a report of a disruption in that gene, but really not a lot of new families found, not a lot of confirmation that those were really bona fide risk genes. And so what I really wanted to do when I started my postdoc at the end of 2009 was to try to implement these new technologies with new approaches to really get, for the first time, at novel single genes of major effect that would likely result in autism when they're being disrupted. And so how we were going to do this is actually to use some new approaches. And so first I'm just going to highlight why kind of the traditional human genetics approaches have been challenging in face of this large-scale heterogeneity. So on the left here, I've shown kind of the classic case control model that's been used in all kinds of studies, including genetics, where you try and get a bunch of affected individuals, which I've shown here in black, and match them with unaffected individuals, which I've shown here in white. And the hope is that by comparing these affected and unaffected individuals, you can learn something about whatever you're studying. And the underlying assumption that you make is that all these affected individuals are affected for the same reason. But it turns out that, let's say in this toy example, that there are 3 different causes of this disorder. In this case control model, your power is going to be rapidly diminished to actually find individual factors that contribute to the disorder. And this is particularly problematic in autism, where we think there's probably hundreds of different risk factors. So the other major approach used in human genetics has been to study these multiplex families. So that multiplex just means that there's multiple affected individuals. I'm showing you here just a toy pedigree of a couple affected children. And if there was just a single gene for that resulted in autism, then this model would work very well, because what you're able to do using human linkage studies is actually combine families. So if you had, for example, 10 families that all had cystic fibrosis, and you combine them together, they would all show signal to the cystic fibrosis gene, and you could add up the signal across the families, even though in any individual family you didn't have enough power. But just as signals would add, they would also subtract. So in that case, if you had 10 different causes of cystic fibrosis, all of your signal would cancel each other out. And so what we've set out to do the past 5 or 6 years is actually to study a different type of family, so what are called simplex families. So these are just families that have a single affected individual that appears out of the blue, with kind of no previous family history of autism or anything related to autism. And what we've wanted to do is actually study in those families new mutations that just have recently occurred in that affected individual. So these would be mutations that just occurred in either the egg or the sperm that resulted in the zygote that led to that individual. So these are mutations that were not present in the parents and have never been present in the family. And our hope is that at least some fraction of those new mutations that have just recently occurred have something to do with autism. And we had, I think, good reason to think that that might be a tractable approach. And I just outlined here 2 examples of why we thought that. So the first here is looking at what I'm calling chromosomal outliers. So these are basically individuals that had visible chromosomal abnormalities that we've been able to see under a microscope for 20 or 30 years, where we had the co-occurrence of a new disorder, in this case in the pedigree A, autism, and a new inversion, so a de novo inversion in chromosome 7 that disrupted 2 genes. And as a graduate student in that SAGE group, we were trying to use this as a strategy of mapping new genes with autism and other disorders. And we'd also known for a long time that in families that looked like this, they had a higher incidence of these visible chromosomal abnormalities. What we also knew more recently, before these studies started, was that when we looked at copy number variations, so these would be large deletions and duplications of the genome, when we look at simplex families relative to multiplex families or control families, we tend to see a larger number of these new mutations in simplex families. And this is specifically talking about these copy number changes, so big, large deletions that delete tens to hundreds of genes. So what we wanted to do moving forward using these exome technologies is to actually try and get at a different type of mutation. So get at basically these new point mutations by sequencing the exomes of an entire family. So sequencing the father, the mother, the affected child, and in most cases we also sequence an unaffected sibling. And the real power for gene discovery in sequencing these exomes is, and finding point mutations, is we actually get down to a single gene. So a single letter of the genome being changed. And so we have a great specificity in knowing that that is the actual gene that is disrupted. And really what made this all possible was the efforts of the Simon Simplex collection to actually go and recruit a large number of families that fit this profile of having a single child affected with autism and an unaffected sibling and no previous family history. And so we had kind of the intersection of new technologies, new techniques, and also new cohorts. The other nice thing about studying the exome is we only expected, before we started these studies, to find about 1 mutation in the exome per generation on average. And so if these types of new mutations actually play a role in autism, we actually won't have a lot of noise to look through, because we'll just have about 1 mutation per child per family. And then we could also take advantage of the fact that mutations should occur randomly throughout the genome. So you shouldn't see mutations of the same genes over and over by chance if those mutations have nothing to do with what you're interested in studying. And so the flip side of that is that if we do see recurrent disruption of a gene or recurrent mutations of the same genes over and over again, that might be a new way of actually finding new genes that are high confidence risk factors for autism. So really this is the entire strategy in a nutshell that has really led to this rapid acceleration of gene discovery in autism. So now for the rest of the parts of this section, I'm going to briefly tell you about some of our exome studies that we've used as a kind of unbiased genome-wide way of finding new candidates, and how we then went and rapidly validated many of these new candidate genes using a targeted resequencing technology called molecular inversion probes, or MIPS. So first, let's find some new novel candidates. So what I'm showing you here is just a summary of 2 studies that we did. The first couple studies with Evan Eichler and Jason Dury looking at sequencing trios, so the father, the mother, and the child, in these simplex autism families. And so in 2012, we had sequenced in our group about 209 of these trios and about 50 unaffected siblings. And we were able to actually find that one needle in the haystack, the one new point mutation, pretty effectively. We found about 1.3 of these mutations per proband, which is just the affected child. And one of the things that we noticed right away is that you can actually figure out which copy of the genome these mutations occurred on by looking at other variation that is transmitted from the parents. And so we're able to actually link the mutation event to some of these other pieces of genetic variation that are traveling in the family. And we were able to do that for about a third of the mutations we found. And we found a very strong bias for those events occurring on the paternal genome, so presumably occurring in the generation of the sperm that led to that individual. As I said, we found about 1.3 mutations per child, so we had literally hundreds of mutations, and many of them intersected very interesting genes that you could make a story about potentially being involved in autism. But in terms of finding recurrent mutations, so finding more than one mutation in a single gene, we actually had 2 genes that had exactly 2 mutations each. And so we were able to infer from that there's going to be a lot more than 200 single genes that result in autism when disrupted. And most of you probably know that we weren't the only ones doing these types of studies. And 2012 was really a landmark year where many hundreds of families were sequenced using this type of approach. And the first thing that you might have been wondering reading those papers is, did we find anything that was actually related to autism? Did we find any autism genes? And I would say at that point, I think the answer was maybe, but we're not really sure. So we had over 900 parent-child trios sequenced all together. And when you even combined those 900 families together, we still had a very small number of genes that we found recurrent disruptions. So in fact, we only had 6 genes where we had 2 or more of what I'm going to call these likely gene disrupting mutations. So those are the types of mutations that we would predict would be most likely to impact the function of a protein. So these would be nonsense mutations or early stop codon mutations, canonical splice site mutations, or small insertions and deletions that disrupt the coding of a protein. And so those 6 genes are probably pretty good candidates for likely being new autism genes, but I think many of us were still skeptical about how many of those would really validate and would we really find additional mutations in those genes. As I mentioned, we can do some kind of modeling based on how often we find recurrent mutations, and based on that, all the groups estimated that there were likely hundreds of genes involved in autism, just based on how often we see recurrent mutations under this kind of very strict sporadic disorder de novo mutation model. But I think looking at these various studies, we had lots of hints that this isn't just a random set of de novo mutations, that they may actually have something to do with autism. So I think first off, I just wanted to highlight, I'm only going to highlight a few aspects of these various studies. 2 that I'm going to show here deal with network analysis, and that's basically just looking at how a group of, in this case, proteins interact. And so one of the things we did is we took kind of known protein-protein interactions from a large database and said how often do our genes that are mutated interact with other genes that are mutated. And we were very surprised to find is shown here on the right, which is this large, highly interconnected network of proteins. And we call this a beta-catenin chromatin remodeling network, largely because beta-catenin was a very well-known gene and very highly connected. That's been known to be important in development, and the fact that we had lots of chromatin remodeling proteins in the network. But we still don't really understand what this network might mean biologically. So I think an important point to make is that this could just be the network of the most mutable proteins in the genome. This might have nothing to do with autism. So one of the things that we tried to do is take a list of genes that had at least had some prior suggestion they might be involved in autism and ask, how similar do this new list of genes that we've newly discovered look in the context of a network space? And so the simple way of thinking about this is, picking a random person off the street and asking, how often do they happen to be connected to you through like a Facebook network? And so what we're really surprised to see is when we did this type of analysis, that the new mutations that we found were actually in bulk shown to be really highly statistically significantly similar to previous autism candidate genes, but most of that signal actually only came from these connected genes that formed this network, suggesting that this network has something to do with autism risk. And then I think another really kind of seminal observation was made by the Cold Spring Harbor group was that many of these genes that we find new mutations in happen to be downstream targets of the Fragile X mental retardation protein, suggesting that that proteins that gene regulates may form a common pathway in autism. So I think at the end of 2012, we were all wondering what would come next, and could we actually not just identify new candidates, but try to find all the new candidates for autism. And so 3 of the groups kind of joined forces to sequence the remaining samples from the Simon Simplex collection, which led to this publication in 2014, where we had data on over 2,500 children affected with autism and about 1,900 siblings. And with this very large data set, we were able to actually, I think, show definitively that, that in bulk, these mutations do have something to do with autism. And I'll just share 2 pieces of data that suggest that. So the first is when we looked at the comparison of the rates of mutations between probands, so affected children in red, and unaffected siblings in green. And what I've shown, what's shown on the slide here is kind of increasing levels of severity of mutations. So, for example, if you look at synonymous mutations which don't alter their protein sequence at all, we basically don't see any difference between affected and unaffected children. We actually see a statistically significant difference of about 7% between probands and siblings for missense mutations. So these are mutations that alter a single amino acid in a protein. But what you can appreciate from the height of the green bar here is that there's a big background of these mutations. So there's many of these mutations in the genome just by chance. So that makes determining which of these individual missense mutations that are found in kids with autism are actually meaningful somewhat difficult. And so what we've done and others in the field have done is focused actually on these more damaging mutations, so these likely gene disrupting mutations. And when you look at that difference, you actually see that kids with autism have almost twice as many of these likely gene disrupting mutations than their unaffected siblings, suggesting that both that they're a major component of risk in these families, and also that we're going to have a much easier time sorting out which of these mutations are actually related to autism relative to the missense mutations. And so what we were able to do in that study is actually kind of look at the contribution of all of these different types of mutations, these new mutations occurring, by comparing the different rates in the affected and unaffected children. And so when we add those all up, for all the 2,500 families sequenced, we see there's about a 28% likelihood that in these families, there's one of these new mutations that's actually contributing to autism. And that includes both these large copy number mutations, these likely gene disruption mutations, and these missense mutations. And the other really intriguing finding that we had, albeit with many less girls than boys in the study, is that it seems that girls may actually have a much higher mutation load. So if you just look at the girls in the study, about 45% of those have one of these new mutations. The other interesting finding is that there's a higher rate of these mutations as well in boys that were on the kind of lower half of IQ of the cohort. So all of these data suggest that these mutations aren't just random and they have something to do with autism. So to make a very long story short and many papers condensed into a single slide, at the end of 2014, we had about 1.2 of these new mutations, point mutations per child, with strong parent of origin bias, where most of the mutations seem to be occurring in the genome of the father. We estimated there's hundreds of risk genes involved with autism under these mechanisms. And we were starting to build a list of moderate to high confident mutations, specifically focusing on these likely gene disrupting mutations. So at the end of 2012, we had about 6 genes. At the end of 2014 in the SSC, we had 27 genes, and combining with other data sets from the Autism Sequencing Consortium, 39 genes. So we're really moving into a place where we're starting to get to the point where we have high confidence novel genes for autism. As I mentioned, we saw enrichments for genes that are involved in modifying chromatin, which is responsible for how DNA is packaged and how genes are turned on and off. Association with genes that are regulated by the Fragile X syndrome protein, and also, which I didn't talk about in terms of time, genes that are expressed embryonically, specifically in the embryonic brain. So as I like to say when I've given this talk before, we still have work to do because we really want to get to single genes that we have ultra high confidence in, so that many of us will spend the rest of our careers, our talent, our time, our treasure, figuring out what they actually do. And so one of the ways we thought we could potentially do that quite rapidly is actually moving to a targeted resequencing approach. And so what really facilitated this happening quite rapidly was some work we did to develop this really ultra cost-effective way of sequencing a rather modest number of genes relative to the exome, so 50 to 100 genes, but in a very easy to do and cost-effective manner. And so shortly after our paper got accepted in 2012, we started to kind of go after our top 44 genes in basically all the samples we had available. So just to remind you, we had sequenced about 200 families, and we had about 200 different candidate genes. We picked 44 of them, and we sequenced them in really a matter of months in basically the entire Simon Simplex collection and a few other samples. And this is, to me, still pretty amazing, given that as a graduate student, I sequenced about 2,000 genes in 6 years. And by finding all the new mutations in those genes, we were able to actually start to ask this question of, do any of these genes have anything to do with autism? So we actually found 27 new mutations in these genes in 16 of the 44 genes. They're actually very skewed towards likely gene disrupting mutations, suggesting they may have something to do with autism. Almost all of them fell into that protein-protein interaction network that I showed earlier, suggesting that has some potential relevance. And importantly, we started to see a lot of recurrence. So we started to find 3 and 4, and in the case of CHD8, 7 additional of these likely gene disrupting mutations in a single gene. So this was in addition to the 2 mutations we had found by exome sequencing. So that's all well and good, but then we were left with the question of, was any of this more likely than we would have expected by chance? And so what I'm showing you here is a couple different Poisson distributions for different genes. And you might imagine that gene A is maybe the largest gene in the genome or the most mutable gene in the genome. So even finding 3 different mutations in this gene would not be that unlikely by chance. And so we really wanted to know, for these specific genes that we were looking at, how likely should we have been to find recurrent mutations in these genes, and if our findings were outside those expectations, use that as a new way of implicating autism risk genes. And so to do this, what we did is actually relatively simple. So we just defined a relative weight for every gene in the genome using some population genetics approaches, so that for all of these 20,000 genes in the genome, we had some idea of what their relative mutability should be. And then we could kind of just spin a weighted roulette wheel based on those weights and randomly place mutations all throughout the genome, and do this for 2,500 people in our kind of 44 genes of interest, and then look at the results specifically in our 44 genes of interest. And so when we did this, we actually never found in all of our simulations, which in this case was 500 million, 27 of these new de novo events in these 44 genes. And so what I'm showing you here is a graph that shows the negative 10 log of the p-value. And so the higher the bar, the less it was found in our simulation. And likewise, we actually never found 7 mutations in CHD8. And actually, in the simulation, on average, we should have probably only found around 6 de novo mutations in these genes, not 27. And so you would probably already believe that CHD8 was probably going to be a real risk gene for autism, but what about the rest? So we can actually draw kind of a multiple testing correction line for all the genes that we were studying, and actually show that these 6 genes meet statistical significance and should be considered at varying levels of confidence for being involved in autism risk. So one of the really things I really like about this study is that we actually have real p-values associated with these different genes. So you might not want to go and start your lab studying TBL1 XR1 until we have some more data, but you'd be very confident in moving ahead with studies of CHD8. And you can kind of decide on what your tolerance or risk is. And importantly, just these 6 genes actually represent a very large, a large fraction of the Simon Simplex. So about 1% of the kids in the Simon Simplex collection have a mutation in just one of these 6 genes. So what we decided to do next, after this paper came out, is maybe we could actually, again, quite rapidly go after a large set of genes that have been implicated as candidates from both these exome studies of autism and also intellectual disability. And so we've kind of plowed ahead and did 64 more genes in even more samples. And I'm just going to go through this quickly because I've already introduced the topic. But we sequenced about 3,500 programs and 2,500 siblings using this MIP approach. And what we were able to show in this data set is that we actually have a couple interesting observations. So one is that we have more of these likely gene-disrupting mutations in these 64 genes in kids with autism versus unaffected siblings. It's actually 9 times as many, which is pretty amazing. By doing these larger sample sizes, we're actually able to get 3 or more mutations in 8 different genes and got up to 14 different mutations in CHD8. And by using the same weighted gene mutation approach, we're able to actually show 9 genes in this study that reach significance, including the novel ones that are shown here in bold. Also interestingly, many of the genes that we've now shown to be significantly mutated in autism are actually mutated in other studies. As I'm showing here 2 of the top genes in encephalopathies, CHD2 and SANGAP1. And combined, just these 9 genes actually represent about 3% of all the autism cases that we sequence. So in conclusion, I hope I've shown you here today that, and in this this new observation, that many of the genes that we've actually found are actually mutated across different developmental disorders, and we could probably spend a whole webinar on that topic. This de novo paradigm has been extremely powerful and high yield. In just 5 years, we've now have 50 to 60 of medium to high confidence risk genes for autism, which I mean is pretty mind-blowing to think about. But I think the sobering thing is that there are actually hundreds of more genes left to validate or find. And many of these genes are going to be mutated in less than 0.01% of kids with ASD. And so that's really prompted the need to increase our cohorts and our ability to find these recurrent mutations in genes that might contribute to less than 1 in 1,000 cases of ASD. And so it looks like I did a good job on time, so I have a couple minutes to tell you about some new frontiers in autism genetics. And specifically, I'm going to just briefly tell you about some new efforts to increase cohort size and what those might lead to. Touch a little bit about whole genome sequencing and how that's moving ahead and will give us new insights compared to exome sequencing. And then hopefully spend a couple minutes showing you about cryptic variation and some of our unpublished work. So I think many of you know that a very exciting initiative launched last Thursday called SPARK. We're really excited to be one of 21 clinical site partners participating in this effort. And the goal for SPARK is to recruit 50,000 families in just 3 years using these local and national partners. Many of the samples are going to get whole exome sequencing and genotyping done. And this is really going to give us the power not just to find new genes, but to actually validate them with high confidence, even genes that are only responsible for, I think that should say 0.01%, not 0.1%. And we're really going to be able to also start to explore the roles of other types of variations in these genes, so inherited variants in particular, both rare and common. And I think most exciting for me is to try and actually learn what these different potential genetic subtypes of autism look like at the phenotypic level. And can we use that information to learn anything about the pathophysiology of autism? And and including developing biomarkers that can be used in clinical trials. I think the other exciting thing that we're going to be hearing more and more about is genome sequencing in autism. I've just shown you here this really beautiful Circos plot from a recent paper from the Sabat lab, where you can see the millions of different variants, both inherited and de novo, of different flavors that are present in genomes. And on the right, you can see kind of the ever-increasing numbers of families that have been sequenced as part of these studies. I think near the end of 2016, the beginning of 2017, we're going to be at a point through projects like Missing and efforts from Safari to sequence the whole genomes of the Simon Simplex collection, the point where we were in 2012, where we're starting to have close to thousands of families that have whole genome sequences and start to really look at whole genomes in a way that has enough power to actually discover new things about autism genetics. But I'd say that there are still going to be challenges. So relative to the exome, the genome has 50 to 100-fold more point mutations, and that's just considering this one class of new mutations. And it's really going to be really difficult to know the impact of these point mutations on the non-coding or regulatory parts of the genome. So there's just still not great ways of predicting that. I think we still have limited spatial and temporal data on the different regulatory elements that are active in human brain development. And importantly, I think we still really need to think carefully about new ways, computational ways of how to model the combined effects of all this variation across the genome. But I think there are kind of several exciting prospects to dealing with these challenges. I've shown these here. So one is genome editing. So with these new Cas9 CRISPR systems, you can kind of quickly insert or remove any mutation you want in the genome. So maybe that will be a useful technology at getting at the function of these elements. And as far as knowing where and when to look in the genome, we're actually starting with network approaches, starting to see convergence into different parts of the brain and periods of development. And so maybe those will help us kind of hone in on the different genes and regulatory elements that control them. And then I think importantly, we're starting to get new models. So specifically, I've highlighted here induced pluripotent stem cell models, where we can actually try to mimic in a dish these spatial and temporal periods that might be important in autism development. But I just wanted to highlight that even with whole genome approaches, we're still going to be missing things. So one of the things we're going to be missing is complex structural variation. I've just shown a nice plot here from a paper that showed this very complex paired duplication that's flanked by this large inversion. And this is a type of thing that we'll just never see with whole genome sequencing, and is probably playing a role in some part of autism risk. And so we really either need efforts to incorporate these types of technologies, like these large insert jumping libraries, or a kind of a fourth generation sequencing technology that gives us much longer reads so that we can sequence through these really complex regions. One of the things that our group has been actively studying is somatic mosaicism or post-zygotic mosaicism. And these are basically new mutations that occur not in all cells, but actually later on in development. So in this case, I'm showing one that occurs just in the ectoderm. And what makes these challenging is instead of being in a single copy in all cells, they're in a much smaller allelic fraction. Most of our technologies aren't really tuned into finding these variants. And so one of the active areas in development in our lab is trying to study these somatic mutations through various stages of development. And as a first pass in that effort, we've been, I'll skip this slide, but we've been looking at the mutations that appear in the Simon Simplex collection. And so what we were able to actually do is go and take a closer look at all of the new mutations that we had previously published in the Simon Simplex collection, and actually show that about 4% of them actually appear to be mosaic, and that they have a highly shifted mutation frequency from where you would expect, where basically it should be 50-50. And so what we've been doing now is actually doing a systematic evaluation of these mutations throughout the entire SSC, using a combination of a custom genomic workflow and validation at large scale using these molecular inversion probes. And so I thought I would just show you one last slide where we have actually looked at these different mutations at kind of decreasing coverages and increasing mutation frequencies. And this is just because you need to sequence, many more times a piece of DNA to detect a mutation that's at a very low frequency. And so what we've seen in the first almost 400 families that we've analyzed is actually this interesting trend for missense mutations where we actually seem to be seeing more of these missense mutations in probands versus their unaffected siblings, but it's not all missense mutations. It seems to be only these that are at a higher mutation frequency. And so one hypothesis for why this could be the case is that maybe what we're seeing is that in the probands with autism, more mutations that are occurring earlier in development, because the earlier a mutation occurs in development, the higher mutation frequency it should have. So with that, I'm going to end on my acknowledgement slide and just let you all know that there are many people that could not fit on the slide. Most importantly, I wanted to thank the patients and the families for taking their time to be involved in research, and thank all of our various funding sources, post and present.
Great, thank you, Brian. We just have time for a couple of questions. We've had a few, so if your question doesn't get asked, I'll endeavor to send them to Brian after the talk. Dustin Baldridge asks, how can we as a field move from interpreting likely gene-disrupting mutations to interpreting missense variants? And similarly, do the findings of de novo variants provide insight into interpreting inherited variants?
That was a great question. So I would say that I think as a field we're taking several approaches to that. So I think one of them is to just sequence more people, and I think that's going to be a challenge to actually have that work. So that thought is that if you just sequence enough people, we'll have enough data and we'll be able to know what's a really interesting mutation for this protein versus not. But what I think we're actually going to need instead is actually empirical data generated in the lab for pretty much all possible mutations of a gene of interest in different types of contexts, so that we have some way of weighting a priori the likely effects of that variant on function. And so that's a pretty challenging effort, but there's actually lots of new exciting breakthroughs in synthetic biology that may actually make that possible, and as I mentioned, including these genome editing technologies. And then as far as the inherited variation goes, I think efforts like Spark will look at not just the simplex families but multiplex families, and by narrowing in on these parts of the genome that we've already identified have strong likelihood in being involved in autism risk, many of those should also have not just de novo mutation risk but inherited mutation risk, and we should be able to look at those in these multiple affected families.
Great. Charlotte De Stefano asked, could you explain how the high heritability of ASD is also related to de novo mutations? She said that she's confused about how the heritability can be so strong for some genes if they keep finding de novo rather than inherited mutations as being key to autism.
Right. That's a great question as well. So one of the ways that heritability is calculated is looking at how often identical twins share a diagnosis. And so in in the case of identical twins, they share both copies of each shares an identical genome, and so if there's a new mutation that occurs, it'll occur in both their genomes. And so they'll still show this high concordance rate if de novo mutations are playing a significant role relative to dizygotic twins. But what makes it complicated in autism is that we likely have lots of different, what we call genetic architectures, all playing a role in what we call autism. So there's these new mutations, these de novo mutations I've talked about. There's likely also inherited variants coming from the parents, which then you would see in the dizygotic siblings in some families, and there's also, of course, environmental factors as well. So as far as I know, there's still not any great epidemiological data on how often families are simplex versus multiplex. Maybe that's something that we can glean from Spark. But deconvoluting these kind of these issues is a challenge. Yeah.
Okay, and finally, Pam Feliciano asks, how many trios need to be sequenced to identify the entire universe of the autism gene candidates until we can move to an MIPS approach to validate candidates?
It's a great question. So I don't have a great answer for Pam. I would think that it all depends on how low you want to go as far as how often it's occurring in the population. So I think once we start getting data on 5 to 10,000 families, we'll start seeing saturation, and we'll stop seeing new mutations we'll stop seeing recurrent mutations that we can actually detect. Doesn't mean that there's not ultra rare causes of autism out there still to find, but at some point, as I think is rightly pointed out by the question, we will stop discovering new things in the exome relative to the cost of actually sequencing them.
Okay, great. I'm afraid that's all we've got time for today. So thank you, everyone, for participating. And just to let you know, a complete replay of this presentation will be available on the Spectrum News site in a couple of days. The webinar is part of an ongoing series, so please check our site for upcoming talks, which will be announced soon, and for video archives of the previous webinars. Any questions that you wanted to ask on this webinar but weren't able to, please feel free to share them in the comments section on the webinar once it's posted to our site, and Dr. O'Rourke will be able to see them there. Okay, and thanks again, Dr. O'Rourke, and thank you all for tuning in.